Hello, welcome to an update on MegaTox. Hi, I'm Sean Eakins, CEO of Collaborations Pharmaceuticals. I have nearly 30 years experience using computational approaches to toxicology and ADME predictions. I've published widely on this topic, over 400 papers and 2 edited books alone on computational toxicology. So what I hope to do today is give you a little overview of where we are with our approaches at Collaborations Pharmaceuticals. Collaborations Pharmaceuticals develops cutting-edge AI approaches that we apply across pharma, consumer product, and the chemical industry. We provide our software for licensing, as well as offering our services for fee-for-service projects with collaborators. As you can see on this slide, we've publicized some of the collaborations we have with various industries. Some of these are shown here. Some of the work is related to licensing our software, and some is related to the fee-for-service projects. As you can see, there's a pretty wide diversity of the types of companies that we've been working with. We've also been able to work on many different types of projects. Heavy focus has really been on toxicity prediction, followed by target prediction. And then obviously we've done work around software development as well as clinical data modeling. Over the years, we've developed a number of machine learning related products. These include key projects, Assay Central. But today I'm going to talk about MegaTox. MegaTox is basically a customizable collection of machine learning tools and models that we've developed over the years. These are obviously focused heavily on ADME and tox issues, and these can be applied to many different industries, as I said, including pharmaceutical, consumer product, as well as chemical industries. Over the years, we've been able to build hundreds of models for toxicity, including leveraging public data sets from Vega, EPA, Tox21, ToxCast, etc. We provide basically the ability for the client to select which models they would like implemented in the MegaTox software, so that we can then tailor the models that are obviously used by that particular industry. Over the years, we've been able to publish the work. This provides additional validation for the models and our capabilities. As you can see here, a number of those papers are in pretty highly ranked journals. An additional optional extra that we provide with MegaTox is the ability to do read across. And we've heavily really focused on this, primarily because we wanted to be able to provide this within a secure environment within MegaTox. So we're able to basically perform a lot of the types of analyses that are available on the EPA's genera. But we've also added the capability to do both classification and regression analysis. And obviously, performing this within the secure environment of MegaTox is a big advantage. So what I'm going to show you briefly is a short overview of the software, and hopefully this will focus in on some of the major points. So what I'm doing here is just giving you a high-level overview of a cytotoxicity model. So you can see different visualizations with the different algorithms and the statistics for the models. On this slide, you can see the ROC curves, and then looking at different ways to visualize that data for each of the individual models. And then we're going to go and perform a prediction with a set of molecules. So we're pointing to a library of molecules that is already uploaded in the software. We're basically asking it to build predictions with all of the different models that I've just shown you. And now it's going to take you through briefly looking at the output of those predictions. So you can see the color highlighting, and then also the different predictions for each of the models. So those were classification models, and now we're going through and looking at each of the molecule details for that set of compounds. And we can also visualize the molecule, so overlapping this particular test set with the training set for this particular model. So I'm just pointing to t-SNE and UMAP visualizations. And so what it's going to do is show you all of the data for the training set and the test set. But you can also focus in on the test set, for example, or in this case, the test set molecules. And then also this is, you can highlight the points. Another thing we can do is do the read across, as I mentioned earlier. So we can select a molecule. We can have options on what fingerprints to do the similarity with, and then run the read across. And what we'll see is the nearest neighbors to the molecule shown on this visualization. And you can click to get a little bit more information about the molecule. And then obviously go through all of the data coming out of ToxRef, highlighting which molecules have toxicity or appear to be okay. And that is a very brief overview of the software. So just to highlight some of the new updates that we've added into MegaTox, some of the ones that I've highlighted here in yellow include a lot of front-end updates that we've performed, especially around the heat maps and visualizing the data. We've also added more features around dataset space visualization. And then we've added some new fingerprinting descriptors. As you can see, we've got quite a few other additional features that we're adding into the MegaTox software as well. One of the things I'd like to highlight during this presentation is that we've published quite a bit of science, new science for us at least, in 2025, shown by some of these images of the papers that we have. And one of our most recent publications is around drug-induced seizures, for example. We've leveraged an adverse outcome pathway to identify 67 different targets, and then we've curated those data sets from Kemble. This has allowed us then to build over 2,000 different machine learning models, and then enable us to make predictions for drug-induced seizure liability. Obviously, this is important for CNS-related drugs because they have a very high level of failure. And therefore, if we could try to predict if there's going to be side effects, that would obviously be advantageous. So obviously, being able to do these very large-scale predictions against targets that would relate to seizures could give you a heads up, across a very large number of targets. And so we've come up with a visualization that tries to summarize all of these predictions with the different models as well. Another recent publication, actually it was just recently accepted, is around steroidogenesis models. And obviously this is an important area because cholesterol is basically modified to produce biologically active steroids. And so if you interfere with any of these steps, that could have a big physiological effect. And so we went about building these models for all of the different enzymes involved in the pathway. And this new publication basically highlights this alongside experimental validation of the models as well. So we've generated some new data and then used that to evaluate the model predictions. In addition, we've built a model for steroidogenesis assay that we've used, and that had about an 80% prediction accuracy. And another example of some recent work that we've been doing is using the MOLBART large language model approach to model P450 substrate activity. And so you can see on this receiver operator curve on the right-hand side, the models appear to be pretty good with five-fold cross-validation using this MOLBART approach, which is basically a state-of-the-art large language model. And in addition, we built in confidence metrics into these models as well. So this gives you a better idea of assessing the quality of the predictions that come out. So you can imagine these models would be used with a new molecule to predict the likelihood that it's going to be a substrate for each of these individual P450s. So that would be of particular interest to pharmaceutical industry. Obviously, we put a lot of effort around adhering to different regulatory guidelines. And this slide gives a little bit of an overview of the OECD guidelines. And so we try to comply with these where possible with the different machine learning and QSAR models that we build as well. So we get asked a lot of questions about what differentiates us as a company. Obviously, we put a lot of focus on the data curation side and also the customization of MegaTox so that the client gets the models that they actually need. And clearly, what I've just tried to show you is that we also put quite a bit of focus around publishing our models and model validation to provide more confidence in what we're producing. There are a lot of competitors in the space around ADME and tox prediction. And as you can see here, I think MegaTox stacks up pretty well against some of the competitors. Obviously, focusing heavily on the customization of the models, having literally hundreds of these already built for the client to choose through is a bit of an advantage, I would think. And then also the software, as you can see, is pretty intuitive and interactive. So providing lots of options around different fingerprints for the models, as well as the model highlighting visualization aspects. And I think also we stack up pretty well in terms of the cost. So additional aspects that I think are important, clearly we put focus on security with the software. And we've been able to provide this on Amazon Web Services, or we can provide it on our own cloud as well. And in addition to what I've shown you today, we do provide customizable support and training. And then, as I mentioned, we have a lot of expertise around fee-for-service consulting. And thank you for your attention.